Ma'am from NEET World. So yes, today we are in our last session where we are solving NSEJS 22 paper with the concept. So today is the last session beta for biology questions of NSEJS 2022. From the next session we will be starting NSEJS 2021 paper. Okay, so far we have dealt with 2024, 23 and this will be the last session of 2022 session paper. Okay, so today we are going to deal with two concepts. One from the microbial world, that is viruses we are going to deal with, both the lytic and lysogenic cycle, and the second concept that we are going to deal is the systems of classification and plant kingdom. Without much delay, let us get started. The very first question, one of the following statements is not applicable to virus. Okay, so in the question he is saying like one of the following statements is not applicable to viruses. Okay. First, option A you see ma, the protein capsid of the virus does not enter the host cell. Yes, you take an example, for example a bacteriophage which has head, collar, tail and tail fibers. This will be adjoined to what? A bacterial cell. Okay, so this tail fibers will be adjoined to the bacterial cell wall, whereas the genetic material, this will solubilize this will contain the lysozyme and it will solubilize the cell wall of the bacterial cell, and the genetic material is deposited into the cytoplasm of bacteria. It is not capsid of the virus does not enter the host cell. That is correct statement only. Second, you see the genetic material is either DNA or RNA but never the both. Yes, virus will have the genetic material, it can be double stranded circular DNA or single stranded circular DNA or single stranded RNA. So, DNA will be in those viruses, DNA will be as genetic material in those viruses which will infect animals and bacteria. Okay, those virus which will affect animal cell or bacterial cell will have the genetic material as DNA. Those viruses like tobacco mosaic virus which infects plant cells will have the genetic material as RNA. Okay, this will have the genetic material as DNA, and those which infect the plant cells will have the genetic material as RNA. Clear, right? So yes, the genetic material is either RNA or DNA but not the both. Second statement is also correct. Next you see the virion replicates autonomously outside the host. Okay, a virus, the singular form of virus is virion. Virion will never replicate outside the host cell. Without a host cell, virus cannot replicate its genetic material or it cannot replicate itself. That is why viruses are not said to be living cells, rather they are termed as nucleoprotein structures or infectious agents or living entities. Okay, so this is wrong. Okay, the virion replicates autonomously outside the host cell, it cannot replicate without the help of a host cell. Fourth statement you see beta, the virus replicates in a bacterial or other host cell. Yes, virus replicates in a bacterial or other host cell. One of the following statements which is not applicable to virus. So, which is not applicable to virus over here? Option C will be our answer. But now in our class we will look into virus, its structural description, different types of viruses, and we will also look into the lytic and lysogenic cycle of the viruses. Okay, so let's get started with viruses. Viruses best described as what ma? Infectious particles or infectious agents. These are also called as living entities or nucleoprotein particles. Okay, they are well described as infectious agents or nucleoprotein particles, or they are also called as living entities. Fine. Next, each virus will contain single type of nucleic acid, either it can be DNA or RNA. Nucleic acids is enclosed in protein coat. Sometimes this is also covered by what? Envelope of lipids or proteins or carbohydrates. So, usually virus will have two components, ma. One is the outer protein coat, inside which you will have genetic material. These are the two components of viruses. What are they? First is genetic material and the second will be protein coat. Protein coat. But in few viruses like HIV, herpes virus, they will be having an envelope over this protein coat. Those are what? Those are having what? Envelope over the protein coat. Envelope can be made up of lipids, proteins or carbohydrates. Okay. Viruses are obligate intracellular parasites. They cannot reproduce unless they invade any host cell. Okay. Use host genetic material. They will use the host's genetic material and metabolic machinery to produce the progeny or to replicate its own cells. They will cause serious damage and diseases in humans, plants, animals and even for the bacterial cells also they are infectious. The scientific study of virus is termed as what? Virology. This is the general information about viruses where we have to know for sure. Next. Viruses do not exhibit most life processes of the cell. Okay, that is why they are not termed as a living cell. Okay. However, they maintain the genetic continuity through multiplication. Through multiplication, they will maintain the genetic continuity. They will undergo mutations, making them more than just inert molecules. Okay. Now, we will look into the structure of viruses. Size ranges, how much will be the range of the size of viruses? Virus size usually ranges from 300 nanometers, for example, tobacco mosaic virus, to 20 nanometers, for example, parvoviruses. Next, they are comparable to small bacterial cells like mycoplasmas and ribosomes. These are small cellular organelles, right? Ribosomes are amembrane bound organelles where you will found in eukaryotic cell and prokaryotic cell. Mycoplasmas are wall-less bacteria which are the smallest living cells. So, its size comprises, we can compare their size with small bacterial cells like this. Next, morphological types. Viruses are classified on their shape and structure into helical viruses, for example, tobacco mosaic virus, polyhedral virus, example, adenovirus, spherical virus, for example, influenza virus, and complex virus, for example, bacteriophages. All the types or shapes of the viruses with example you have to remember. Okay. Next. Yeah, here you see different types of viruses with examples. I have tabulated it, just go through. First is helical virus. How can you describe a helical virus as? These are long rod-like structures. They may be rigid or flexible. For example, rabies virus and tobacco mosaic virus. These two are the examples of what? Helical viruses. Second type is polyhedral viruses. Polyhedral viruses, they will be having many sided shapes. Okay, they will be having many sided shapes. Example, herpes simplex virus and polioviruses. And then we have enveloped viruses. Third type is what? Enveloped viruses. Viruses with capsid enclosed in an envelope. Virus with capsid enclosed in an envelope. This is roughly spherical. For example, influenza virus and HIV. Okay. And then fourth type is complex viruses. These are having complicated structures, especially those infecting bacteria. For example, bacteriophages like T4 bacteriophage. Okay. So, this is the tabulated form of this. Next. Yeah, some viruses like measles have spikes over the envelope. They will be again having spikes. For example, these spikes are composed of what? Glycoprotein projections. Okay, example of having spikes over envelope is measles. Important, remember, that will help in the host cell attachment. Spikes are crucial for virus entry by binding to the receptors on the host cell. Basic components of a virus include two main components. One is the core that contains nucleic acid or genetic material. Second is capsomere, that is protein subunit. Protein subunit. So, nothing but genetic material and protein coat are the two components of viruses. Look into the genetic material, and this protein coat is having what? Capsid. Capsid is the protein coat, and the protein subunits which are present in the capsid are called as capsomeres. Okay, what are capsomeres? Protein subunits that are present in the capsid are called as capsomeres. Next, so here you can see in measles virus the envelope protein projections of spikes, glycoproteins, and envelope inside which protein subunit will be there, and inside that the genetic material is present. After this, yeah, so genetic material can be double stranded DNA, single stranded DNA, or double stranded RNA, and it will be mostly in the circular form only. Genetic material is in circular form. So here if you see, plant viruses usually have what? Single stranded circular DNA. Animal viruses will often have double stranded DNA. Bacteriophages also will have double stranded DNA. Some viruses, for example HIV, have two identical RNA molecules. These are called as what? Genomic copies. Okay, and the shape of nucleus is circular or linear, but mostly it is circular. Mostly it is circular. Fine. So this is all about the genetic material description of viruses. Next. Now we will see the replication of the virus. Okay, virus will replicate only in the host cell, it cannot replicate outside the host cell. So it conducts two cycles, lytic cycle and lysogenic cycle. Virus replication can be as lytic or lysogenic cycle. Fine, lytic or lysogenic cycle. First, what is lytic cycle? If virus particles entering into the bacterial cells, virus entering into the bacterial cell will replicate amongst the bacterial cell. Inside the bacterial cell, all the virus particles will replicate and increase in their number, and then they will burst out the bacterial cell, releasing out the virus particles. So in lytic cycle, this will cause bacterial cell lysis. Bacterial cell death happens through the lytic cycle. Whereas through lysogenic cycle, the genetic material of virus will be integrated into the bacterial genetic material. As such, bacterial cell lysis will not happen. Okay, bacterial cell death will not happen, but the duplication of viruses will be happening. Fine. Whereas in lytic cycle, bacterial cell death or bacterial cell lysis will happen. Okay, clear? Next. So here if you see, yeah. First, attachment of bacteriophage on the bacterial cell. Attachment. The phage attaches on the surface of the host cell. The next is penetration of the genetic material into the bacterial cell. Phage DNA and the host DNA. Okay. So now phage DNA will be replicating, utilizing the complete metabolic machinery of the bacteria. It will start replicating, biosynthesizing what? The protein coats and the genetic material, and they will get matured. New phage particles are assembled, and last it is lysis. Cell lysis will happen, releasing out the newly made phages. This cell lysis will cause what? Bacterial cell death. Clear? This is the lytic cycle. Next is lysogenic cycle. So in lysogenic cycle, what happens? First is, again, bacteria will come, attach, it will sit on the bacterial, virus will come, it will sit on the bacterial cell, and through the lysozyme which is in the tail fibers, the wall will get solubilized, and the genetic material will be injected into the bacteria. As such, first adsorption takes place, then penetration of genetic material. This genetic material will be integrated with the bacterial genome, and then integration happens. After that, replication. Replication happens along with bacterial DNA. Okay, viral DNA will not replicate on its own, it will replicate along with bacterial DNA. And then induction, when the daughter cell is induced, the viral DNA is excised. Okay, and this viral DNA will synthesize new particles, they will assemble, and again the new particles will be released. Okay, direct cell death will not happen over here. So this is what lysogenic cycle. At the end, what we have to say is lytic cycle or lysogenic cycle will be operated by the viral particles inside the bacterial cells, but viral particles cannot replicate outside the host cell. Okay, and what all we have learnt till now with viruses are very important, about viruses are very important. Same the case you have to learn with other microbes as well. Okay, next. So here there are clear differences between lytic and lysogenic cycle. Once go through, yeah. First, lytic cycle, a viral replication cycle that ends with the cell lysis. Lysogenic cycle, a viral cycle when the viral DNA integrates into the host DNA, and this lies as dormant, this is dormant. Next, virus type, here you can see virulent phases, here you can see what? Temperate phases. Integration of viral DNA does not integrate with host DNA over here, but here viral DNA will integrate with the host DNA. In lytic cycle, host cell is destroyed, here host cell remains alive. There is no latent phase over here, but latent phase persists in the lysogenic cycle. Viral DNA replication replicates independently using what? Host machinery. Virus will replicate independently using host machinery. But here in lysogenic cycle, viral DNA will replicate with host DNA as the genetic material is integrated with the bacterial DNA. And then symptoms are very rapid here, symptoms will be low here because direct cell lysis will not happen, and examples include for lytic cycle T4 bacteriophage, for lysogenic cycle it is lambda phage. And then this will trigger for activation, immediate lytic action will happen. Here this causes stress like UV light and chemicals, and this will trigger the lytic cycle. So these are the clear differences between lytic and lysogenic cycle, please go through it. And next, what will be the best reference books to learn about virus, bacteria, its classification, different shapes and sizes, and then the replication modes that are operated in the virus and bacteria. So these are the reference books for preparing topics of viruses, virus classification, general biology in NSEJS. These books should be referred. NCERT class 9th and 10th, where fundamental biology basics will be there. In that, virus, binomial nomenclature and classification of virus is given. Next is Truman's Elementary Biology of volume 1 and 2 you have to go through. Standard reference book is Campbell. Okay, and you can also go through to explore more questions on viruses. You can go through previous year questions of MTG NSEJS Explorer. Okay, now we will look into the second question. This second question is from plant kingdom. Okay, core botany question. This is from plant kingdom. So here you look into the question first. Carolus Linnaeus, a Swedish botanist, is credited with binomial nomenclature of plants and animals. While using binomials, he devised a system of classification of plants. His classification is called as what? So far from plant kingdom systems of classification, we have 3 systems of classification. Those include artificial system of classification, natural system of classification, and phylogenetic system of classification. Phylogenetic system of classification. First you see artificial system of classification. Artificial system of classification is also called as sexual system of classification. This is proposed by Carolus Linnaeus and Aristotle. Okay, Aristotle and Linnaeus have proposed this system of classification, wherein they have classified the plants based on gross morphological characters and the androecium structure. Based on what ma? Gross morphological characters, and based on androecium structure. Fine. Next is natural system of classification. Natural system of classification is proposed by excuse Natural system of classification is proposed by Bentham and Hooker. Okay, this is proposed by Bentham and Hooker, where they have considered both the external characters and internal characteristics of plant, both external and internal characters of plant. Okay. Next, phylogenetic system of classification, which is proposed by Engler, Prandtl, Hutchinson, Taktajan. These are the proponents of phylogenetic system of classification, where along with external characters and internal structures, they have also considered evolutionary relationships. Okay. They have also considered evolutionary relationships. Fine. So, these are three systems of classifications we should be noted with. So, who devised? Carolus Linnaeus has devised what system of classification here? Artificial system of classification or sexual system of classification, that is option A. So, this will be the right answer over here. Yeah. Just look into the theory once ma. Linnaeus from 1707 to 78 his time period, he has introduced binomial nomenclature, where this gives the concept of genus and species. His classification was artificial because it was based on the morphological characteristics like number, arrangement of stamens and carpels. So, that is why we will also call it as sexual system of classification. Natural systems consider many characters, both external and internal. Bentham and Hooker were the proponents. Phylogenetic system of classification consider evolutionary relationships, example Engler, Prandtl. Next, cladistic system is based on shared derived characters and branched sequences. Thus, Linnaeus system of classification is artificial classification. What are the popular books that you have to refer if the question comes from the plant kingdom? For that concepts, what books you have to refer? NCERT Science class 9th and 10th, and then Truman's Elementary Biology, Campbell. So, these will be the standard books all the time. So, these books you can refer for core botany chapters also. So, with this, today's session is done ma. Today's session of NSEJS 2022 paper is done. In our next class, we will look into the NSEJS 2021 paper. Okay. So far with the previous questions, you can go through the playlist of NSEJS biology. Okay. Thank you.